The overall goal of this procedure is to use live cell imaging to assess cell fate decisions following treatment with the anti-mitotic drug Paclitaxel.
This method enables us to visualize how individual cells respond to different doses of the anti-mitotic drug Paclitaxel. The main advantage of this technique is that it provides a wealth of information on single cells or whole populations that is unobtainable from fixed cell imaging or population-based assays. Demonstrating this method is Marc Vittoria, an MD-PhD candidate in the Ganem laboratory.
To begin, clean the glass bottom of the imaging dish with optical cleaner to remove any fingerprints or dust that may interfere with imaging. Use sufficient optical cleaner to wet the entire glass surface. Place the glass bottom dish with cells on the microscope stage in the imaging dish adapter. Remove the plastic cover from the dish and cover the dish with a glass-topped chamber. Ensure the chamber has a valve to allow a steady flow of air consisting of 5% carbon dioxide. To humidify the 5% carbon dioxide, the gas is flowed through tubing inserted into a sterile water bath housing. This allows for the gas to become equilibrated to 95% humidity. Initiate and calibrate the encoded stage using the software program controlling the microscope. This will ensure accurate XY coordinates and prevent focal drift. Next, perform the color illumination by raising the condenser and closing the aperture so that the field is dark. Lower the condenser until a crisp hexagonal shape of light is visible. Then, use the centering screws to center the hexagon of light in the field of view. Once the hexagonal shape is visible,